slow rolling start in case we have a few stragglers. But again, welcome so much to this session focusing on research for SCA27B. If we haven't had the chance to meet, my name is Dr. Celeste Seward. I'm the Patient Engagement Manager for the National Ataxia Foundation, and it's my absolute privilege to be able to host this series. A few housekeeping items before we jump into the talk today. If you need closed captions in order to better understand and interact with the session today, you can turn those on at the bottom of your screen. There's a button with the letter CC on it, and you can turn those on for yourself. And if you'd like to connect with other members of the community who are attending this session, you can use the chat like you are right now to connect and share with where you're joining from today. And just to see a few more that came in, we have people from Virginia, from Caracas, from Florida, Arizona. Hello, welcome. So if you'd like to connect with other attendees, please use the chat feature. If you have any questions for our speakers, though, we ask that you don't put them in the chat. Instead, please use the Q&A window, which you can also find at the bottom of your screen. And that button looks like it has the letters Q&A on it. And with that, it is my privilege to be able to reintroduce our two speakers today who joined us for our first session focusing on clinical care. And now we're going to be discussing what's going on with the research for SCA27B. The first is Dr. Brais, who is the director of the Rare Neurological Disease Group of the Montreal Neurological Institute of the Hospital for McGill University. He follows a large number of ataxia patients in his clinics at the Montreal Neurological Hospital, the Montreal Lucie-Bruneau Rehabilitation Center, and the Saguenay Neuromuscular Clinic. His research largely focuses on genetic basis of neurogenetic disorders with founder effects in Quebec, with a special interest in ataxias and myopathies. Dr. Brais has played an important role in identifying causal genes for multiple disorders, including SCA27B. So that's the most recent one, caused by a GAA expansion within the FGF14 gene. So he's our first speaker, but we have a double act for you today. Our second speaker is Dr. David Pellerin, who is currently a neurogenetics fellow at the UCL Queen Square Institute of Neurology and the Hospital for Neurology and Neurosurgery in London, the United Kingdom. He received his medical degree from the University of Sherbrooke in Sherbrooke, Canada, and subsequently completed his residency in adult neurology at McGill University in Montreal, Canada. And with that, I'll turn the floor over to the two speakers today.
Thank you, Celeste. Next slide. Thank you all for joining, and thank you for NAF's invitation. This is a follow-up from our first talk. Before we start, we're still going to review a few things so that people who are less familiar with this condition can better understand what we're going to talk about. I must say to start that talking about biology, talking about pathophysiology, talking about how to explore how a disease is caused, and talking about how we can test medications are tough subjects to present on this type of format. So please, we'll do our best. David and I will do our best, but if you have questions, please address them. We're going to try to keep as much time as possible for questions to, again, try to review these questions, these elements we're presenting to you. So a little bit more about the spinocerebellar atrophy, type 2B, spinocerebellar ataxia type 2B. So we call it a late onset. So in fact, if we switch slides, this slide shows a young person, but in fact, the people who develop are more people of my age, people who will develop around the 60 a permanent ataxia, and it's a very slow evolving ataxia. So it's quite different than many ataxias that people see in the clinic, or ataxias that are more publicly known, like Friedreich's or others, even some of the SCAs, which will start in their 20s or even earlier. So it's a late onset. It's very slowly progressive. It has a very not unique, but a very strong or very high prevalence of cases that have downbeat nystagmus. So people are told your eyes are beating downwards, and that's kind of guides us to this diagnosis, though it can be found in many other ataxia. The other thing that's very striking is the episodic nature, and that's important when we think of the pathophysiology. The episodic nature, it means that some people get more ataxic symptoms and then they go away, and these period of episodes can last from a few minutes to a few hours, rarely longer. There are many visual disturbances. People can see double. A lot of people wear prisms in their glasses later on. They can have this kind of the image is moving, what's called asilopsia, and a lot of them have visual blurring, which may be just a subtle form of diplopia. There's a lot of vertigo or dizziness in this form of ataxia, particularly during their spells or and particularly early on. And there is also a striking intolerance to alcohol, far more than in other ataxias. In fact, if people will take just a little bit of alcohol and will feel they walk like a drunk, which is not the case for common people. And so let's not change slides already. Can you go back, David? Yeah. So and what's interesting, because we're going to discuss it a little bit later, is this idea that it's slow, early episodic impact on walking, and the using of walking aid is high, but using a wheelchair is very low. And so it's really a disease that had been only marginally studied because of this kind of late and not as strong an impact, though the people who have it don't want to have it, obviously. But it's that there's no wonder that it's a late discovery, and also because it was a harder mutation to find. Next slide, please. So we know now that it's all over the world. There's more than 20 countries that have reported cases, and it's growing every week. I like this study from a group in Spain, because they defined a group of 107 patients with what we used also as a definition, people who start their ataxic symptoms after the age of 30. And as you can see from this group, there's many causes, but the largest known cause is SCA27B. So it's the most common cause of late onset ataxia in this population. Though for the ones who are with us today who still don't have a diagnosis, look at the number they have, more than 43% without a diagnosis. So there's still a lot of work to be done for these late onset ataxias. Next slide. So let's turn to the biology. We know a lot about this. The gene is called FGF14, which is fibroblast factor 14. The name is not really relevant for the conditions. It's a large family, the fibroblast growth factors. What is relevant for us is that there's two forms of what's called isoform. That means, again, that's a complex concept, is that the gene can be read in two ways from the genomic data, and one includes a large intron in which there's that mutation, and that's isoform 1B or isoform 2. And what's striking about that isoform is that FGF14 is very expressed in many tissues, thymus, brain. FGF14, the isoform 1B or 2, is largely expressed in the cerebellum. So when we found the mutation in that gene, it made sense that it would be that could cause this disease. In fact, as we will see in a minute, there was already a form of ataxia related. So if you look at the bottom part of this slide, starting from the left, the dark blue means there's a lot of the gene. And why they look rounded is because those are cells. Those are the famous Purkinje cells. A large major output of the cerebellum come from these cells, and you see they have a lot of blue. That means they have a lot of this gene being expressed. If you look at the right panel, which is what it shows is that we know that this FGF14 gene produces a protein, and that protein will bind to subunits of sodium receptors and even some other receptors. What it means for people with ataxia is the cerebellum is important for balance. We know part of that role is it has a kind of rhythmic function. It's polarized, depolarizes, polarized, and to do that, it has to bring ions in and out in a kind of rhythmic way, and that requires these channels. And the Purkinje cell is doing that all the time, so it requires a lot of those channels. So you can imagine if a protein is less abundant, maybe the organization of these regions where the rhythmicity is happening will be imperfect, or in fact even more sensitive to, for example, alcohol that may disturb the function of those channels. Next slide. So we know also that this variant, or this isoform B, is very, very expressed mostly in the brain, and that is shown on the left side with the entire body, where you see the pink. There's pinks elsewhere, like they show in the lungs, the heart, but the more expression is shown in the brain, and in particular And this is the cerebellum that you see. So really the cerebellum is where you have a lot of FGF14, and particularly the B form, compared to other parts of the brain. And that has to be important for why the cerebellum is more affected and therefore leads to ataxia in those patients. Next slide. So it's very aesthetic. This is kind of a painting, but I think the point for you is that this is a way, if we look at cells from a brain and we try to separate them using different means, and we look at which of those cell population, and the brain can be divided in very simplistic terms in neurons, electrically cells that are important for communications and discharge, and insulation, which is the glial cell, the ones that form the white matter that allow conduction and so forth. And if you split them using these very high-tech technologies, you can see that in fact the white, the yellow in this slide, the glial cell, which is the insulation, doesn't express very much FGF14. However, if you look at the neurons, all the neurons express a lot FGF14. So it's clearly suggests that it's a neuronal disease. So it fits again, rich in Purkinje cell, rich in neurons. Now we still have to work out better why having this error causes a problem, and I'll give you a few elements in a moment. Next slide, please. So what we're going to show now is a mouse where the gene was inactivated, the 2 copies, and that was published in 2002. And you see the mice has this kind of what we call paroxysmal hyperkinesia. They kind of contract and they fall. And so this kind of, it's a very dramatic phenotype. They also have a tremor, which we can see in SCA27B. So you can see the kind of movement of the leg. But the rest of their behavior, which is related to other brain function, is pretty much normal. They will search food, they will move around and do things, but they will have a lot of paroxysmal. So in fact, a lot of episodic events. And that fits the phenotype in our patients, at least the early phenotype. This being said, this is a complete loss of the 2 copies of the gene, which is certainly not what we have in this condition. Next. So just a few words about SCA27A. We did not decide on the name of the disease. It was decided by a body that said, no, there's already a condition, there's already an ataxia called SCA27A that was described in the early 2000s, which is a disease which is transmitted from a parent to half of their siblings. So it's a dominant disease. It has earlier onset, more in the 20s and even earlier in some cases. Yes, it has nystagmus, yes, it has tremor, but it has also a lot of neuropsychiatric and intellectual disability. So it's quite different from what we were seeing. And the mutations are illustrated in this, in the sense they're usually single errors in the coding sequence. Next slide. So what we found is an error in an intron. So it's in red there. So the intron it doesn't code for the protein. As I said, for SCA27A, the error is in a region that codes for an amino acid in a protein. This is not the case. So what we discovered, and that was discussed in the first talk, is that we see different expansion of this region. This region will grow in size is larger in these families. It's a little bit somewhat difficult, and I saw that Selma from Chicago is on the call today, who's really developed the first platform for clinical diagnosis in North America. Thank you for attending today. And we are, with her, dealing with this idea that we don't know everything about the expansion and diagnosis. What we know is if you have 300 and more, and it's pure GAA, you will have the disease. If it's 250 to 300, we don't know. We don't know, meaning that in some families, some people did manifest the disease, some didn't. And now there's another category, which is the uncertain, the people with 200 to 249 size. We're still working it out, guys. We still don't know. So it's a problem for you getting this information, but we still don't know. It's a question of time. We'll be able to tell you and develop other ways to be sure. If it's less than that, which is more than 98% of the population, then you never develop it. So there's a lot of variation. there's mixed information in the literature, and that's the graph on the left side, whether the size will influence severity. We know if you get 2 copies of an area, you'll be more severe, start earlier. If you have a larger expansion, we don't know. There seems to be a trend, but it's difficult. So please, in your family, if your other members have a higher size, it doesn't mean that they will necessarily have a more severe disease than you, or inversely, if you have a bigger size, you will be more affected than others. So there is probably a trend. It is subtle, and it will take time to be able to give a complete answer to that. Next slide. So what we found, and that's important for understanding disease, and is that, in fact, if you look at the level of the gene has that area. If you look at the level of the RNA, there's less of the RNA, particularly the 1B, or the second isoform. And at the level of protein, there's less protein. So there's a relative haploinsufficiency. That means there's less of the protein. And that's not only in the brain of patients, it's also when you take, in this case, fibroblasts, and you use a special magic and you make them into neurons. Those neurons are not able to pump out a lot of the protein and a lot of the gene. So it seems to bring down the expression. Remember the mouse we saw before doesn't express it at all and has a very dramatic phenotype. Our patients are pretty normal early on. It catches up with episodic, and then it starts to be permanent. Next slide. So the impact on the brain is that the brain, some areas, as now you know, the cerebellum expresses a lot, and the Purkinje cells express a lot of this gene. The neurons, the Purkinje at one point, that's what's illustrated, will be depleted. You will lose them. So you see what my colleague is showing is you should have a lot of those brown dots on that slides, but there's a lot of them missing. And with time, part of the brain, particularly the central part, which is called the vermis, will shrink a little bit. And that we can see on the MRI. And that's the most classical manifestation on the MRI. But many ataxia can give you that. So we know there's kind of a vulnerability of the cerebellum, and it's probably related to the fact that this gene is so important because it codes for protein that's important for the function of the cerebellum. Next slide. So if we talk of the biology, so we need to, so what do we know about we know that it's not a fast evolving disease. So someone looked at one marker of degeneration, which is called NFL, neurofilament long chain. You can use that to see that the brain is aging quickly and cells are being destroyed, and it goes into the CSF, so the liquid that bathes the brain. And what you can see in the left border is that in GAA, FGF14, there's not more than in controls. However, in, let's say, a more severe neurodegenerative disease like multisystem atrophy, then there's a higher level. And you can see higher level and has been followed in different conditions. This was done in FGF14, and you can see, based on age on the right panel, you can see that it's all over the place, and it's not very different from normal people around the same age. So it's clearly not, and we knew that from the clinic. Next slide. That it's not rapidly evolving. So how do we study? And some of the slides are provided by NAF. So some of you may have seen many of those those talks, and it's kind of helping you to get an understanding of how this hard work is being done, and hopefully also to make you aware that this takes time. So at this point, we have access to you. We have access to generous patients who have left their brain for research. So that's been a very important material in my laboratory and a few others in the world. We still don't have a model. The model that I described, we showed earlier, was the mouse with no gene. This is not what we have in patient. They have one good copy and one with an expansion. So there's new models being developed, mice, rat, by different groups. And my group is not doing this at this point. So those are being developed, and I'm looking forward to the results and having others share this information. Next slide. But they're needed, Then we can do a lot more, and NAF has encouraged that in many projects, is to develop all types of resources that can be shared: blood, plasma, CSF, skin biopsies that can be transformed and pluripotential cells. And again, postmortem. So those are all the tools that, or resources that any group who studies ataxia can use. Next slide. So we can also look at patients in many ways, looking at natural history, looking at different imagery, looking a lot at eye movement, which are very perturbed in this condition, using neuroimagery, not only to understand which part of the brain are affected, but also and helping therefore diagnosis, but also using them maybe as an outcome measure in studies for therapeutics. And then we use all the tools of the laboratory, the functional studies and the genetic studies. In our case, we work a lot with a colleague in this hospital who's a specialist in white matter disease and ataxias. Her name is Roberta Lapiana, and in the context of a large international collaboration, we're trying to pull together imagery from many, many groups to try to really get the best possible image of how it evolves in time imagery-wise. Next slide. We're showing some data. So the data is saying that, in fact, what you see in the cerebellum, it's largely the vermion or the central part that is affected rather than the other parts of the brain. And on a single metabolic study, which is called a PET scan, you can see that there is a loss at lower panels here. You can see that there's less signal in these vermion. Is it only related to loss of volume or also clearly loss of activity, which would go with the idea that these patients have, that patient, because it's a single patient, has lost some of its very active, metabolically active cells, the Purkinje, that's why there's less perceived activity. That will need more confirmation. Next slide. So there's also a way to help researchers doing natural history studies. Many groups are following those patients already. NAF has included SCA27B in the one of the conditions that will be included in the CRC SCA in Montreal. It's Antoine Duquette at the CHUM that will do this follow-up. The NAF clinic is there. I'm a member also of this clinic, but it's headed by my good colleague Antoine Duquette. There's also the AGI will try to, which has been endorsed in July by the Ataxia Global, will try to help refine what would be followed. The AGI is focused on what is the best, preparing for a clinical trial. So it really will be helping the people who want to do large natural history. It doesn't have the resources to do large natural history per se, but it will be a place where researchers from all over the world can share notes, ask questions with the data they have, and certainly influence how we follow patients to have the best outcome measures to accelerate therapy, confirmation, and discovery. Next slide. So just a few words about natural history. We already know a fair amount from different studies. This is a very good study from the Tübingen group, where, again, they're very hard to follow for you, but I think there's maybe 2 points I'm going to make. If you look at the what we call the spaghetti plot, which is the one that has many colors, so those are individuals who evolve in time. So you see the flatter the line, the less it evolves, because this is time. Horizontally it means time. And then the other scale is the SARA, which is the degree of effect. And you see that there's a lot of variability. And even if you went to the details, you see that there's a lot of up and go, because patients are different from one day to another, and also some of it is observer. So it's a very slow evolving disease. If you look at the panel below, which is saying, when do you start using, based on the evolution of the disease, when did you start using different walking aid? What's interesting is the violet at the bottom, which says that, in fact, even after 25 years of the condition, there's very few people who will use wheelchairs. And that, I think, is one of the major questions we're asked in the clinic when people come in their early 60s, my age, 62 again, and they've had an ataxia for 3 years. They have a vague or very definite family history, and they say, well, look, am I going to be in a wheelchair in 3 years? We can already know, if you can confirm that, we can tell them it's unlikely. It's not only unlikely in the next 3 years, it's probably unlikely in your lifetime. And that by itself is very reassuring. Again, there's variability, and we're still learning how to be as clear, but still precise, and not as precise when the information is not there. So I think it's slowly evolving, and the other are more complex to explain. But the impact on speech is also very slow. The impact on hand function is also slow and not really large. balance is more affected, and there's no strong evidence that cognition is affected in any way. And remember, the little mouse did very well without the gene functioning wise and organizing their daily life, but without the gene, our patients are clearly very bright and highly functional people, and we don't see an effect of that. Next slide. So I'm going to turn now to David Pellerin for the therapeutic approach. And before having him talk, I have to thank him again. David has been a pillar of this project, and also the person who produced the nice slides for us today and last time. So David, the stage is yours.
Thank you, Dr. Brais. And so to conclude this webinar, we're going to go over some of the therapeutic approaches that are now being studied in SCA27B. As Dr. Brais mentioned, because SCA27B was only recently described, there still are no disease-modifying therapies under development. And that is simply because the community has just now started to study the underlying biology and pathomechanism of this newly described disease. However, the pace at which the field has been moving forward over the last year is actually incredibly fast, and we're actually quite hopeful that disease-modifying therapies will be being developed in the coming years. But nonetheless, through close work and collaboration with Matthias Sanofsik from Tübingen in Germany, it rapidly became clear to us shortly after the description of this disease that 4-aminopyridine, as you'll see in a minute, which is an already marketed drug used to treat multiple sclerosis, among other conditions, appears to be a highly promising symptomatic therapy for this condition. But just before talking about 4-aminopyridine, I just want to say a word about acetazolamide. Some of you may know that acetazolamide has previously been shown in clinical trials to be effective to reduce the frequency and severity of episodes of ataxia in episodic ataxia type 2, which is caused by mutation in the CACNA1A gene. And because SCA27B is also an episodic ataxia, we wanted to study whether this drug, acetazolamide, was just as effective in this patient population as in patients with episodic ataxia type 2. And so what we did is we performed a retrospective open-label real-world multi-center studies enrolling 34 patients from Quebec, Innsbruck, Austria, Perth, Australia, and Nancy in France. And we specifically looked at whether acetazolamide reduced the frequency and or severity of episodes of ataxia in patients with SCA27B as judged by themselves in a non-blinded way. And what we found is that 56% of patients with SCA27B treated with acetazolamide actually had no benefit or no response to the drug, whereas 6% had a non-sustained response, which essentially faded away in a couple of days or weeks. Only 38% actually had a response, and in the majority of the patient, this response was actually quite mild, so not a great response. And I should point out that a few patients had actually episodes of ataxia triggered by this medication. And so one should be cautious when trying acetazolamide, as provoking episodes of ataxia may be a side effect of this therapy. But now talking a little more about 4-aminopyridine, which, as I mentioned early on, appeared to be a promising symptomatic therapy as we initially reported in our initial studies earlier this year. And that was really thanks to the work of Matthias Sanofsik and his colleague in Tübingen, Germany, where upon discovering SCA27B, they went back to their cohort of patients and they looked at patients with SCA27B who had been previously treated with 4-aminopyridine. And among them, they found that 7 patients that were now diagnosed with SCA27B had been treated with 4-aminopyridine. And in fact, when they looked at these 7 patients, they found that 6 of them actually benefited or had a response to 4-aminopyridine. And this response was rated as marked for most of these 6 patients. And the drug basically reduced the frequency and severity of their ataxic symptoms. And so these preliminary studies actually prompted them to perform 3 additional prospective N-of-1 treatment experiences in 3 patients. And here I'm just showing you the results for one of these patients. But basically what they did is they gave the medication to the patient. They looked at their symptomatic time per day. They kept the drug for a few days, and then they stopped the drug. They looked at the effect on the symptomatic time per day. And they did that, in a few cycles. So here, 3 cycles, and they did the same for 2 other patients. And interestingly, what they observed is that there appears to be a tight relationship between symptomatic time per day and drug intake, whereby basically when the patients are given the drug, the symptomatic time per day appears to rapidly drop, whereas when the drug is taken out, the symptomatic time per day appears to re-increase to pre-medication level, basically before these patients took the medication. And this tight association was replicated in 2 additional patients, suggesting that indeed this drug has a promising symptomatic benefit. And when they actually looked at which specific symptoms were improved by the drug, they found that diplopia, dizziness or vertigo, dysarthria were the 3 most improved symptoms, whereas hand and coordination and gait impairment did not appear to respond just as well to the drug as these 3 symptoms did. And interestingly, when they looked at the number of days with severe symptoms in patients on and off the drug, they found that when patients were actually on the drug, they had a significantly lower number of days with severe symptoms compared to when they were off the drug. And so that was certainly a very promising first study looking at the effect of 4-aminopyridine in this patient population. And our German colleagues actually did a follow-up study to look at the effect of the drug on real-life gait performance in a single individual with SCA27B. And to do so, they actually used 3 body-worn inertial sensors on both feet and the lower back, as you can see here in the image. And they monitored 4 different parameters. These are namely gait speed, lateral step deviation, stride duration, and harmonic ratio. And what they found is that all 4 parameters actually improved while on the drug and worsened when the patients were taken off the drug. And what's quite interesting here is that stride variability and harmonic gait ratios are actually 2 parameters that have been previously associated with a higher risk of falls in patients with ataxia. And these 2 parameters actually improved with 4-aminopyridine in that single subject study, suggesting that perhaps 4-aminopyridine might also decrease the risk of falls in patients with SCA27B through its beneficial effect on gait performance. And what I would also like to point out is that the SARA score, which, as Dr. Brais mentioned, is a commonly used rating scale in the clinic, actually failed to capture the 4-aminopyridine-driven gait improvement noted in this patient, whereas these sensors captured it in all 4 parameters monitored or studied. And so this study actually suggests that these digital motor gait biomarkers may be sensitive, meaningful, and ecologically valid outcome measures for future clinical trials. So now, after these 2 promising studies, we went on to look at the response rate to 4-aminopyridine in a large cohort of patients with downbeat nystagmus. But before presenting these results, I just want to say a word about how common are FGF14 GA repeat expansion in patients with or previously diagnosed with idiopathic downbeat nystagmus. And by idiopathic, I mean patient for whom acquired, secondary, or genetic cause for downbeat nystagmus had not been previously found. And so what we did in this study, in collaboration with Mathis Sanofsik, again from Tübingen, and Michael Strupp from Munich, Germany, we took 170 patients with idiopathic downbeat nystagmus and screened them for the FGF14 repeat expansion. And what we found is actually that almost 50% of them carried an expansion, suggesting that indeed this expansion is a very common monogenic cause of downbeat nystagmus syndrome. But now looking at the response at the 4-aminopyridine response rate in those patients, to our greatest surprise, what we observe is that 81% of patients carrying an expansion, so patients with downbeat nystagmus carrying an FGF14 expansion, had a clinician-reported response to 4-aminopyridine, whereas this figure dropped to 31% in patients without an expansion. And clinician-reported response was defined as the physician essentially assessing the patient after 1 to 3 months of treatment, and assessing them both in terms of gait performance. So was the gait improved, the gait parameters improved with the drug, and during ocular motor examination. So was there an improvement in the intensity of the downbeat nystagmus or other ocular motor signs? And when patients themselves actually assess whether they responded to treatment, almost 60% of the patients with an expansion mentioned having had an improvement with the drug, whereas this dropped down to only 11% in patients without an expansion. And again, the patients assess whether their gait was improved and whether they had any improvement in their visual symptoms, focusing on things like diplopia, ortholopsia, and visual blurring. And I should also point out here that all clinical assessments that were recorded as part of that study were recorded before the discovery of SCA27B. And that's simply because those patients received the drug before that expansion was ever described. And so all assessments were naturally blind to the underlying genotype. And what was quite striking is, in fact, a positive response to 4-aminopyridine, either assessed by the physician or the patient, predicted positivity for the FGF14 expansion in patients with downbeat nystagmus with an 89% positive predictive value. And so what these results mean is that that expansion appears to define a subgroup of patients with ataxia or downbeat nystagmus that seem to be exquisitely sensitive or responsive to 4-aminopyridine treatment. And so next, we looked at the functional disability of patients treated with 4-aminopyridine while being on and off the drug. We had that data at hand for 7 patients. And what we actually found is that all 7 patients had a functional disability level that was invariably lower while on the drug compared to off the drug. And I just want to point out here this example here, which I think was the most dramatic example we have, where the triangles represent being on the drug and the circle off the drug. And this lady in particular, when she was taken off the drug in an attempt to try to wean her to see if there was any effect, she actually deteriorated quite significantly, going from walking independently with minimal symptoms to needing a walker or a four-wheeler. And so that challenge was done 4 years in a row, and it resulted in the exact same thing 4 years in a row. And we observed this, not as dramatic though, for 6 other patients. And just lastly, for 4-aminopyridine, as part of this study, we were lucky enough to work with Michael Strupp, who had previously led a randomized double-blind placebo-controlled crossover trial of 4-aminopyridine in 27 patients with downbeat nystagmus. And these patients were randomized to receive either the drug, so 4-aminopyridine, or placebo. And these 27 patients actually had very heterogeneous diagnoses. And so some of them had secondary cause for their downbeat nystagmus, some had other genetic diseases such as SCA6 or episodic ataxia type 2, for instance. But fair number of these 27 patients also had so-called idiopathic downbeat nystagmus. And if you recall what I said a few slides ago, we now know that roughly 50% of patients previously diagnosed with idiopathic downbeat nystagmus now carry an FGF14 expansion. And so presumably, a number of these patients included in that study with idiopathic downbeat nystagmus might very well carry a repeat expansion. And we were fortunate enough to actually recover DNA from 4 of these 27 patients, 4 of which had idiopathic downbeat nystagmus. But just before showing you the results of that, I just want to point out a very astute observation that the authors made back then in this study that was conducted in 2013. Actually, at a group level, looking at the conclusion of this study, the authors found that 4-aminopyridine actually improved the intensity of the downbeat nystagmus and also improved walking in patients with downbeat nystagmus. But what was quite interesting is when they performed subgroup analyses, they found that the largest effect of the drug was observed in patients with idiopathic downbeat nystagmus. And so we actually hypothesized that perhaps these patients who responded the best to the drug, those with idiopathic downbeat nystagmus, perhaps carried an FGF14 GA repeat expansion, as as I've shown you in the previous slide, as these patients seems to be highly responsive to that drug. And so interestingly, among the 4 patients for whom we recovered DNA, we shown that all 4 of them actually carried an expansion. And when we reanalyzed the results from that randomized controlled trial, we actually have observed that all 4 of them had an improvement in the intensity of their downbeat nystagmus while on the drug, but not while on placebo, therefore providing another layer of evidence for the potential symptomatic benefit of that drug in this patient population. Now, although all the results I showed you so far appear quite promising, it is important to point out that these need to be replicated and validated in a prospective randomized controlled trial. And so I know efforts are underway to launch clinical trials. So stay tuned as we hope to that some of these trials will start in the coming months or years. And so what Dr. Brais and I have showed you today is obviously in large part because of your involvement of individuals with SCA27B. And you've been instrumental in helping us better understand this disease and moving the field forward. If for some of you who are not already involved in research, you'd like to participate in research projects or drug development, I'd encourage you to get in touch with your local neurologist who can direct you to clinician or researcher working on this disease. And you can also get in touch with the Ataxia Global Initiative or the National Ataxia Foundation, who are actively involved in SCA27B research, and they may direct you to researchers or a clinician working on that disease. So on that, I'd like to thank you all for your attention, and we'll be happy to take questions.
Thank you so much for a very informational, jam-packed presentation. A reminder to all of our attendees, if you have questions for our speakers, we ask that you put them in the Q&A window at the bottom of your screen, and we're going to get to as many as we can over the next 17 minutes or so. And we already have a number coming in. So we'll start with this one. We have an attendee who's interested in what is the difference between functional studies and genetic studies? Could you tell us a little bit more about that?
Yes, it's a good question. So genetic studies we're looking at how differences in the genes may influence the presentation. So we're really focused on the genetics. So in a way, for example, FGF14, we find the genetic cause, but maybe there's other genetic factors that influence the severity. So that would be a genetic study to look at modulators of severity or even having some protective role. The functional studies are looking at what impact those changes in the genetic sequence have on the function. First, at the first level, the function of the protein. In this case, it doesn't seem to alter the function of the protein, but it probably makes it less available. And that has an impact on function. So that's also a functional study. So a lot of good research, a lot of promising research is when they have a model that allows you to see the impact of the genetic errors or differences on a function that can be measured, whether it's a level or even a metabolic function or survival. And that can go from the cell all the way to a complex organism, which may be a very small organism like a fish, for example, or all the way to a primate. So that's the general terms. It's not many functional studies include some genetics, and some genetic study will have some degree of functional validation, we call. So they're often together, but that would be the way I would distinguish the two.
Fantastic. And our next couple of questions are about the GAA repeats within FGF14. So we have one attendee write in that they were tested with a repeat of 239 for FGF14. Their doctor said that it was too close to rule out that they have SCA27B. Could you provide any additional information on this?
Yeah, so I'll make an introductory comment, and maybe David can also can add something. So we're still working on it. So I think unfortunately, if you're between 200 and 300 or 299, the answer may not be absolute yet. I think if you are in a family, let's say, and you are in a family, we have that, and we're looking at those family at this time. So you are in a family where clearly some people have larger size mutation and develop the disease, and you have a smaller mutation like a 239, but in fact you develop the same symptoms as the others. It's likely causal. So I think it's how are we going to add some layers to make in that case you have the disease. Again, you may be 239 and you may have the predisposition, but you will never develop the disease. So you need to have symptoms. And if you have symptoms, then it's maybe easier. Then the problem is, and I'm sure all the diagnostic labs are going through that, you test someone, there's no clear family history. And then someone has a 239, and what do you do with that? Then you give an answer we don't know yet. And then hopefully through a different other means, imagery, even other things that we're trying to develop as criterias, maybe the combination of that 239 with other elements will say, yes, you have this condition, and therefore that's what you suffer from, that those are the new therapies coming, and yes, that means that there is a true risk for your descendants and other family members. David, do you want to add something to that?
No, I think you summarized it quite well. I think the answer is not as straightforward as a yes or a no at this early stage. I think we have to be nuanced. It's obviously a rapidly evolving field. I think as of now, the absolute cutoff is 300, as you pointed out, Dr. Brais. The 250 to 300 are sort of incompletely penetrant, meaning if you have such an expansion, you may or may not develop the disease. And for lower sizes, I think it's a combination of things that will help us determine whether or not these are causal, SCA27B. And I think you summarized that quite well. I may just add that in that downbeat nystagmus studies that we perform, what we observe quite interestingly is that a significantly greater proportion of patients with downbeat nystagmus actually carried such such intermediate allele, quote unquote, of between 200 and 249 compared to a large control data set. But also not only were these intermediate alleles enriched in these patients, but they also seem to have a phenotype or clinical features that were quite similar to individuals with larger expansion. And so, again, it's not yet clear cut. I think this is rapidly evolving, and we hope to provide you with a more definite answer in the near future.
Then another question about GAA repeats. One of the attendees has read that Friedreich's ataxia is also caused by an intronic GAA repeat expansion. Does that mean that drugs that work for Friedreich's ataxia, could they potentially work for SCA27B? Is there any connection there?
So I'll take a first go at that, and then David, you can add something. So yes, that was one of the amazing thing for us discovering this error, is saying, well, this is the second true GAA repeat, and it's an ataxia, and there's so much work that's been done on Friedreich's. Yes, so maybe some of the symptomatic approaches or treatments that maybe are more generic could be useful. Yes, so that's an avenue, but even more interesting and potentially more promising is the drugs that are being developed to really, I will use a term between quotes, attack that repeat, which is obviously curating a problem. And yes, our impression is that and we don't have strong data at this point, but clearly people who are pioneers and doing cutting edge research on these type of genetic base or molecules that affect the repeat in Friedreich's are really interested and are already studying SCA27B. So I think at this point, symptomatic treatment with 4AP is certainly on the horizon, but maybe also the use of new therapies that have been designed for Friedreich's may apply to SCA27B. So it's really a time of great hope for all of us. David, you want to add something?
No, I think you summarized it quite well.
Okay.
All right. And then our next question is from attendee about what, if any, information is available about why some SCA27B patients have symptoms much earlier than others. So this attendee is sharing that her onset was at 28, but her grandfather had onset of symptoms around 50. She's a bit confused by this.
Yeah, so again, it comes to, I'll let David answer that one. I'm sure he can do a good job on it. David?
Yeah, I think this is probably just as puzzling for us as it is for you. I don't think we have a great answer yet as to why, you know, there's such a large variability within the same family. We think that, you know, perhaps the size of the expansion explains a little bit of the variability, although as Dr. Brais showed you, this relationship between size and age at onset doesn't seem to be as strong as in other repeat expansion diseases. Obviously, individuals who carry 2 expansion may also develop disease earlier. And so that, you know, that is certainly a possibility. But the other possibility as well is that perhaps for some reason that we don't quite yet understand, some individuals will develop episodes of ataxia early on, whereas other might not. And, you know, perhaps you who started at 28, you experience episodes of ataxia, which tend to occur, you know, earlier than the development of the progressive ataxia. And perhaps your grandfather, you know, only developed progressive ataxia later on without the preceding episodes. And so that, you know, that might be another possibility. But yeah, bottom line is we don't know what factors make that, you know, some will develop it much earlier in the family than others.
But perhaps once more people get tested for SCA27B and enroll in natural history studies and there's more data, we might have more information about why there's this variability.
Yeah, and
Yeah,
we're trying to
that's
do
correct.
that. We're trying to do that with AGI Global, the study group. In fact, we'll need cases from all over the world to build enough to kind of come to conclusions on those. So I think it is a collective effort from family members and affected individuals and the clinicians and researchers around the world to share their data.
We're all in this together.
Yeah.
And then the next question, we talked a lot about 4AP in this presentation. We have a number of questions from folks asking about dalfampridine, who they've been prescribed by the neurologist. Is there anything known about how SCA27B patients, the impact of dalfampridine in terms of on symptoms? Is there any information known about that?
Yep. So dalfampridine is essentially a very similar molecule to 4AP, which is marketed as fampridine. In fact, 4AP crosses the blood-brain barrier, which is basically the barrier that insulates your brain from the rest of the body. So 4AP more easily crosses the blood-brain barrier compared to 3,4-diaminopyridine. Which is marketed as dalfampridine. But there have certainly been reports of individuals with SCA27B responding to dalfampridine. And so we're not expecting a marked difference between both, except that perhaps that 3,4-DAP might not cross the blood-brain barrier as well as 4-aminopyridine. So that's essentially the extent of what we know so far. But again, it hasn't been tested at a large scale, and so I don't have a clear answer as to whether, you know, that effect will wean out over time or is less marked than 4AP. But certainly, we've heard of individuals, you know, responding to 3,4-DAP.
That's good to know. We had a quick request if we could stop the screen share so that folks can see our faces a little bit bigger for the remaining Q&A session. Thank you for that. We also had a number of questions about 4AP. Is there any information available, because we know this is very new and we're collecting information together, about if people of different ages respond better to 4AP who have SCA27B, or if there's other factors about how people respond to it?
Can go ahead, Dr. Brais.
No, the challenge that I have no first-hand experience. So I think we're learning from the experience of others. A lot of it is retrospective. Prospective is building. So I can't answer that question really. David? Yeah,
I think you know that's the bottom line. We don't have enough experience to tell whether or not age or other factors have an impact. Nor, you know, if the disease is more advanced, if you've been progressing for longer, will you be less, you know, likely to respond? We don't have that granularity to answer that question yet.
Mhm. So something that may come along with time, but at this point, no clear answer. And then just scrolling through the questions here, we have a question from someone who's interested in learning more about how they can get tested for SCA27B. At least for folks in the US, if you go to the National Ataxia Foundation website, we have instructions that you can share with your clinician on how to order a CLIA-certified test for SCA27B. But for our attendees who are in Canada, how can folks receive genetic testing for SCA27B?
So at this point, many of the tests are sent to Chicago also because it's a clinically accepted test. We do still do it in my lab as a research test. Again, it's not the same report that's generated. I think the data is probably as good, but I think if it's easier to get the shipment done elsewhere in Canada if you get the Chicago-based test. In Europe, there's starting to be labs that are doing it also. So I think it's just a question of time. It's not one of the hardest repeat to test, but it's not one of the easiest either. And so we're all learning, and it's a community. So we share, all the labs that are doing diagnostics are sharing, and we're learning. And it's remarkable that we're less than a year post publication, and we are where we are in terms of expertise and clinical testing. So yes, so I think people can find an answer wherever they are in the world, but they may get two types of reports depending. But I think most of us are using the same techniques and are in fact sharing our difficult cases, trying to come to an agreement as to the interpretation. David, do you want to add something?
No, I think that was a very comprehensive answer.
And how much progress has been made over the past months since the discovery of SCA27B, the gene, has been tremendous. That it's truly a testament to work by yourselves and other researchers around the world and the collaborative efforts in terms of sharing information. It really helps out folks like us on the patient and family side of things. And then we have a couple of questions around things that trigger episodes or that could make symptoms worse. So someone is interested if caffeine can trigger ataxia symptoms if you have SCA27B.
So David, you want to take that one?
Yeah, sure. I mean, this is certainly one of the triggers that's been reported by some of the individuals with SCA27B. It's not the most common, but it's been reported with greater frequency than you'd expect by chance, I suppose. As Dr. Brais mentioned, I think the two most common triggers are alcohol and physical activity, but caffeine's certainly been reported. And things like, strong emotions and stress have also been described by some of the patients. Although how common that is, we still don't have a study that looked at this systematically. But yeah, I would say for that individual who mentioned caffeine, yeah, it's been reported.
And you touched on a few others. People were asking about alcohol. They were asking about exercise. We had someone here who's interested if there's any recommendations or resources for how they can exercise and stay healthy without triggering an episode. Is there anything available on that?
I wish I knew. I think it's calibrating your own abilities. I don't know. What we see in some patients is that they kind of train themselves. They don't accept that they get more ataxic or with activity, and they kind of push themselves through that kind of. I don't know if hydration helps. Some do tell us that they hydrate themselves a lot before. I don't know, and hopefully we will have ways to give stronger indication. But I think the old tricks of hydration, not just water, but probably some beverages that have more than just water, and listening to your body. And some people stop. They clearly tell us they're walking their dogs. They know they're going to get to the park, and then they'll have to stop for a while. And then they start again. So I think it's that, and the sensitivity to exercise is variable. So it's not because a family member is very sensitive to it that you will necessarily be very sensitive, and therefore you should not be doing exercise. And I don't think there's any evidence that doing exercise will accelerate the condition. So really, it's finding the right balance for you as an individual, and hopefully. We don't know that. We don't know, but maybe 4AP, for example, is in fact a good protector for the exercise. induced ataxia. And maybe that will be one of the big impact for some patients, is that they will take the 4-AP before doing their major exercise and then not have the symptoms. And that would also be a great quality of life type of impact.
Well, that would be huge. And it's still very early days, but things are hopeful on the horizon. And with that, we are at time. So if everyone can join me at your individual Zoom screens, thanking both of our speakers, Dr. Brais and Dr. Pellerin. Thank you so much for sharing this with our community and chatting with us today. We really appreciate your time and all the efforts you are continuing to do for the SCA27B community. And for everyone who's listening here today, I hope you found this session helpful and informative and gave you a little bit more information about SCA27B. So wherever you are in the world, good morning, good afternoon, good evening. Hope you have a wonderful rest of your day. Take care, everyone.
Bye-bye. Thank you.